Recently I switched labs and I'm now a postdoctoral fellow in the group of Professor Daniel Engel in the immunodynamics, also at the University of Medicine in Essen. Well, I think one of the things that you have to keep in mind when designing such experiments is that the data quality is crucial in order to really make the biological conclusions that we are looking for. And actually Miltenyi has the right systems for this, especially when thinking of the MACSima system, which I'm a little bit more familiar with. I have the impression that the progress that you made there on the hardware side and on the software side is really astonishing. I'm currently at the University of Medicine in Essen. I recently switched groups, so originally from training I'm a B-cell immunologist. I started off in the lab of Professor Artur Kibler, who basically does research on both sides, so both on the lymphomas and also on the normal B-cell counterpart. And by understanding the normal B-cells, we get a deeper insight into the biology of lymphomas. And my project there was in the beginning the role of human marginal zone B-cells. And over time it developed more into the direction of the general human splenic immunity. So what you need to know about marginal zone B cells is that they are very important in certain types of infections. When it comes to bloodstream infections, marginal zone B cells are crucial. I think it's the combination which makes, for example, the MACSima so exciting for us, because the deep phenotypic characterization, we can perform this using flow cytometry or also using, for example, single cell analysis. But this gives us insight into the single cell, but not into the context where the cell was originally positioned. And using spatial multiplex imaging, as MACSima provides, we can also combine different modalities. So if we, for example, perform multiple types of analysis, so single cell sequencing of the bulk of splenic immune cells, and combine this then, for example, with the MACSima, and especially when thinking of the new technology of RNA-Sky, this might be a possibility to actually link these different modalities. Because if you think of typical analysis of single cell data, that you then look for differentially expressed genes, but most of the genes that you get there, they do not do you the favor that it's a nicely expressed cell surface molecule or whatever, so you can easily identify your population back and flow. But even there, you will be limited. You can isolate the population and do analysis on the population itself, but you are not able to find it back, for example, in the histology and really tell, is this population actually important in the biological question that you have or not. And using RNA-Sky, I could imagine that you can design a custom panel of differentially expressed genes from your single cell analysis and then find back your population actually in the histology and then study its role, for example, in the immune response or tumor biology or whatever. Yeah, the very first time it was actually at the instrument where we just looked at the pre-staining of our marker of interest to define the regions of interest. It was just the overview of CD21 expression, and it was really impressive how just in the, I think it's twofold magnification there, you can just see the region that you focused on for a couple of years now in your research, and it's just right there. I mean, the CD21, it's a very interesting pattern because the red pulp is basically negative, and then you have your spots of very bright follicles where the CD21 is strongly expressed on the surface of follicular dendritic cells, and then there's a small rim of marginal zone B cells at the periphery of the B cell follicle. I assume I will stick to spatial analysis, but I would like to combine it with different modalities, as explained earlier. For example, single cell analysis really gives you deep insight into the biology of single cells, and if you can combine this with a supervised approach, as for example in MACSima, where you can stain for certain cell types and activation states, for example. I think this is one way to even get a deeper insight into what is actually going on there. I think the way Miltenyi does support the scientific community is just right. I would wish that they keep up the good work. Maybe also invest some more time into making, for example, this new technology RNA-Sky in combination with MACSima even more advanced. For example, actually I'm not sure what the limit is for the number of RNA probes that you can use on one sample, but if you can, for example, push it to the same limits as for protein antigens, that would be great.